So we're moving on now to the electron transport chain. We're leaving the citric acid cycle and all that came before it in the dust here. Please don't keep in mind that we are continuing on with glucose metabolism. That story will take this lecture and the one after to complete. This is chapter 20 in the textbook, however, the first half of chapter 20 to be more specific. And we're just going to use this first brief chunk to briefly review the citric acid cycle from a little bit of a different perspective, and more importantly, take a bird's eye zoomed out view of the citric acid, of the electron transport chain in metabolism in general. I want to set the stage pretty carefully for all of you so that the citric, I keep saying that, so that the electron transport chain has its own unique context. The electron transport chain can be fairly confusing if you just dive in feet first, but if you have a general understanding of where you're going in this chain and why, I find that the pieces click in much more readily. So we'll just use this first chunk to get a general, fundamental understanding of the electron transport chain, and then we'll fill in the details with later chunks. This will also be a 3-chunk series here for Lecture 15. As we move on to the next chunks, we will talk about reduction potentials, go back to redox reactions, and look at them just a little bit more specifically, and yes, a little bit more mathematically. Nothing too intense there. We'll finally see, from a delta G perspective, what redox reactions are all about. And then in that last chunk is when we'll talk about the details of the electron transport chain themselves, and you'll be well prepared for it at that point. So by now, I'm hoping there's a great deal of familiarity that you all have with glycolysis and the citric acid cycle. Out of glycolysis, we made a profit of 2 ATP molecules, but more importantly, we made pyruvate, 2 pyruvates, for every 1 molecule of glucose consumed. That pyruvate was used to make acetyl-CoA. That was due to the PDC's actions for us. That acetyl-CoA was fed into the citric acid cycle. Carbon dioxide was made as waste along the way, 1 molecule of carbon dioxide by the PDC, 2 more by the citric acid cycle. That's the carbon dioxide we exhale. 1 molecule of ATP was made directly from GTP in the citric acid cycle. But the question here becomes, why did we really do all that? Did we really do all that for 3 ATP molecules? What was the main purpose of going through these steps of glucose metabolism? What is it that we want to make most from the citric acid cycle? And the answer, hopefully, that you've just shouted at the top of your lungs, loud and clear, is electrons. It's the electrons that we were after here. It's the electrons that were important. Electrons that we harvested from glycolysis, from the PDC, and most notably from the citric acid cycle. We have reduced a total of 12 electron carriers with all of these steps taken together, totaling 24 electrons, which we have captured. What do we do with those electrons? Why did we go through so much trouble to get them? Well, that's going to be the focus of this lecture and the one that comes after it. Believe it or not, one more lecture after this one, and we're done with the metabolism of glucose entirely. In fact, once we're done with glucose metabolism, we talk a little bit about how we store and metabolize glucose in different ways. We have one quick dough tip in the pool of metabolizing amino acids, and then we wrap up with photosynthesis. So the end of the course is actually coming pretty quickly as well. We spent 8 total lectures, or we will have spent 8 total lectures on glucose metabolism. It's such a central story in biochemistry. I've told you many times already in many of our previous lectures that when we are harvesting electrons, what we're really after is energy. That electrons are energy carriers, and it's the energy in those electrons that we're after. The question then becomes, well, where is that energy? How is it that electrons hold energy? And maybe more importantly, how do we get energy out of an electron? What do we do in order to release that energy? Well, that's really what aerobic metabolism is all about. The citric acid cycle, the electron transport chain, and most importantly, oxidative phosphorylation, which is the process that actually makes ATP, is probably the most efficient way to extract energy from food yet evolved on the Earth. All aerobic metabolism occurs in the mitochondria. Everything we've been discussing from pyruvate on has been mitochondrial, and this includes the electron transport chain. Anaerobic metabolism is much less efficient. We really don't make a lot of energy out of that, And we've already talked about it. That's when we can either ferment, one way or another, the pyruvate that we've made through glycolysis. We do that largely to recycle oxidized and reduced NAD and NADH plus H. That's all cytoplasmic. So let's take a little bit more of a detailed look of the organelle that we're going to be doing this process in so that we understand the lay of the land. We are, of course, in the mitochondria. The production of ATP in the mitochondria by aerobic respiration, more specifically by oxidative phosphorylation, is endergonic. The reason why I have obviously there is because making ATP takes energy, just as breaking ATP releases energy. That's why that should be obvious. We bust open ATPs to release energy, which means we trap energy when we make ATP. The energy that drives this ATP production process forward comes from electrons, quite directly. It comes from the electron transport chain. Oxidative phosphorylation and the electron transport chain are coupled processes, where the energy released by the electron transport chain is the energy that feeds endergonic oxidative phosphorylation. So just as an energy-releasing reaction can feed an endergonic reaction, this energy-releasing process of the electron transport chain feeds the energy required to make ATP through oxidative phosphorylation. In a very, very brief and oversimplified nutshell, here's how it works. The electrons that we just harvested from glycolysis, the PDC and the citric acid cycle, are passed from one protein complex to another through individual redox reactions. You can think of this passing of electrons as a chain of electron passers. Well, let's say that a little bit more efficiently. You can think of this process as a chain of transporting electrons. Well, and we just named the process. So the electron transport chain is nothing more than that, a chain of electron carriers that are passing electrons between each other. And here's a very simple version of the electron transport chain, and we see some things that are already familiar to us. We see NAD, that's what's holding some of our electrons right now, most of them in fact. We see FAD holding some of our other electrons, and we see oxygen, which is going to be the ultimate electron acceptor, where all of the electrons end in the chain. Excuse me. The rest of these are just different electron carriers that are holding on to the electrons briefly before they're passed to the next member of the chain. The catch is that each and every time the electrons are passed from one carrier to another, energy is released. It's almost like a game of hot potato, where the hot potato is a battery, and each time that potato lands in someone's hands, energy is released as a result. Or maybe it's hot pink flamingo. I don't really know. But anyway, if anything is passed between carriers, each time that thing is passed, energy will be released a little bit if that thing being passed is electrons. And as we already know, electron passing is called redox reactions. We're very familiar with those. So we've got all the tools we need in place already. We can kind of sum up this whole thing as a chain of redox reactions allows electrons to be transported from carrier to carrier, and each time those electrons change hands, energy will be released as a negative delta G. It's that energy that will be trapped and then used to make ATP. Well, we can handle that. That's pretty simple. So electrons are passed from one complex to another, releasing a little bit of energy at a time, essentially to avoid what I refer to as a metabolic explosion. Too much energy released in one spot, in one place, can be catastrophic. There's a lot of potential energy right now in this diagram. This man is holding a lot of weight up against gravity. That's a lot of potential energy. If he were to decide to release that potential energy all at once, turning it into kinetic energy, that would be by letting go of the bar, there's a major problem. He's going to kill himself. Instead, he's going to slowly release that potential energy in very short motions, slowly lowering that bar back into its stand or into the arms of the spotters. By releasing that potential energy slowly, this gentleman will not kill himself and avoid the kinetic energy explosion of just dropping that bar in place. In much the same way, if all of the energy contained in the electrons we harvested were to be released all at once, that would be too much energy for the cell to contain. It would literally destroy the cell. Instead, just like lowering this bar slowly and carefully, the energy in those electrons is released slowly a little bit at a time so that the cell can capture it and use it in an orderly fashion. That released energy, doled out in small bits with subsequent redox reactions, the redox reactions of the chain of electron transport, is going to be used to pump protons against their concentration gradient. These protons will be pumped across the inner mitochondrial membrane. Now, what did I just say? Yes, the energy that we release from the electrons we harvested will be used to do nothing more than pump protons against their gradient. We will end the electron transport chain with a proton gradient across the mitochondrial membrane. Why we do that will quite possibly be the most amazing and awe-inducing aspect of the life sciences that you've ever heard of before. But I don't want to spoil it, because that's what the next lecture is for. Not the next chunk, but the next lecture, lecture 16. Essentially, though, when we end the electron transport chain, we're going to have pumped a lot of proteins out of the mitochondrial matrix and into the inner mitochondrial space. That's all we're going to do. All of that glycolysis, all of the work of the PDC, all of the work of the citric acid cycle, and indeed all of the work of the electron transport chain itself, all of it was done to do nothing more than pump protons. And that is active transport, pumping protons against their concentration gradient. This is going to result in a proton gradient across the inner mitochondrial membrane. Those protons are going to be very, very unhappy in the inner mitochondrial space. They're going to want to get back into the matrix. That is going to be stored potential energy, energy of wanted motion, and it's that energy that we will capture along the way to ATP production. But that's all I'm going to say about it, because I want to wait for the big reveal in lecture 16. This process in general, the coupling of released energy from the transport of electrons and using that released energy of redox reactions to pump protons against their gradient to create a concentration gradient that will eventually be used to make ATP, that's called chemiosmotic coupling. But we'll come back to that idea in the next chunk. Our goal right now is to just discuss what happens with those electrons. All of those electrons that we have on NADH plus H and FADH2, what do we do with those electrons? All those electrons that we stole from glucose and glycolysis, the PDC and the citric acid cycle, what do we do with those electrons? All of those electrons that are holding all of that energy, waiting to be thrown from carrier to carrier in a game of hot potato, what do we do with those electrons? And that is the story of the electron transport chain. Where those electrons go, protons will follow. We're going to use that to our advantage. And eventually those electrons will end up on oxygen. Protons will follow them, creating H2O. Along the way, we'll regenerate the oxidized forms of our electron carriers so they can go back to the citric acid cycle and pick up another load. But I'm getting a little bit ahead of myself, a little bit too much into the details. I just want to hit you with the overview here, that we are going to have a series of redox reactions, each with small containable negative delta G's, and that released energy will be used to pump protons. That's what we're after. So we hope you leave this chunk with an idea that electrons are going to be passed from one carrier to another, creating an electron transport chain, and that chain will release energy. That released energy is going to be used to pump protons, and those protons will form a concentration gradient that will eventually be used to make ATP. That idea is referred to as chemiosmotic coupling, a term that we will return to in the next lecture. But if you've got those general ideas for now, you're all set to jump into the electron transport chain in the next two chunks.